The broadcast is now starting. All attendees are in listen-only mode.
Good morning, everyone. Thank you for joining us at the latest installment of the HCSA Research Webinar Series. Today I'm really delighted to have two special guests joining us from Teva Pharmaceuticals, Dr. Michael Hayden, who's the President of Global Art Research and Development, as well as the Chief Scientific Officer of Teva Pharmaceuticals, as well as Spiros Papapetropoulos, who is the Vice President and Global Head of Clinical Development in Neurodegenerative Diseases. Spiros is a new addition to the Teva team, joining them from Pfizer, where he was working on their PDE10 inhibitor for Huntington's disease. So we're sad to see him go from Pfizer, but excited to see him stay in HD and be working at Teva. So before they get started, I just wanted and talk about the PRIDE-HD study. I just wanted to go through some of the ways that you can ask a question in the webinar for those that are new to the GoToWebinar platform. So in the control panel on your right-hand side of your screen, you should see a section that says questions. You can just click the plus button and maximize that, and you can type your question for Michael or Spiros at any time during the webinar as it pops into your mind, and send it, and we will ask the questions on your behalf at the end of the webinar. Typically, we put all of these webinars on our website so that they can be viewed at your convenience. Usually it's 6 a week, but we recognize that this is kind of an odd time. We apologize for some of our West Coast community members that might not be able to attend. So we are going to accelerate this and try to get this webinar up and out by the end of the day, and we will certainly make everyone aware of when this archive is available to be viewed. You can also view it on our website at hcsa.org/researchwebinar. And finally, just want to remind everyone that you'll be receiving email notification of our upcoming March webinar, which will be Dr. Beth Thomas from the Scripps Research Institute. Well, she'll be talking about HDAC inhibition as a potential treatment for HD. So look out for that. So with that, I want to turn it over to Dr. Michael Hayden, and I will just go ahead and switch you over, Spiros, as the presenter.
Well, thank you, George. Thank you, everybody. It's a real privilege to have this opportunity to talk with you, as I have in the past. I want to just say hello to many of you who I probably know personally, and to be so close to the families with Huntington disease today, and to share with you what we think is some interesting new perspectives on a trial that we're conducting. Also, this is the first time that Spiros and I are doing this together, and actually Spiros has only been at Teva Pharmaceuticals a short while, and I know many of you know him, and we're really thrilled to have Spiros as part of our team, and Spiros is playing a key role in the clinical development and trials in Huntington disease. Well, the exciting thing today is that we're talking to you, and that Teva is very committed to Huntington disease. We are in the middle of 2 trials right now, one on PRIDE-HD using pridopidine, and another on using laquinimod in HD that has just begun. And if we go to the next slide, of course, my own involvement with Huntington disease goes back some time. And Spiros, if you could just push, yes, thank you, goes back a long way to my time as a young medical student in South Africa almost 40 years ago, where I first saw patients with this illness. And I must say today, what we're seeing, it was just a dream then, that there would be pharmaceutical companies interested in Huntington disease. This was disease unknown and unheard of in South Africa. but really in many parts of the world, people were stigmatized, discriminated, and left to cope with the burden of the illness in isolation and in despair. And it was at one of these meetings as a medical student, next slide, that of course I met the founder and one of HDSA, Marjorie Guthrie, and Marjorie became a source of great inspiration to me, and you can tell that this is some time ago, because even though you can't see me, I'm a lot less hairy now than I was then. And Marjorie became a real source of inspiration, and really was my introduction to HDSA actually more than 40 years ago. So really quite amazing. And of course my own journey, next slide, took me from Cape Town to Harvard Medical School and Children's Hospital and the Brigham and Women's, and then UBC and now Teva. And you may wonder about that journey. And the journey was really a journey really committed to firstly learning more about Huntington disease, doing fundamental research, and then going to UBC, developing the first predictive test for Huntington disease, and then always keeping in mind that our major goal is to do something to alleviate the burden and to reduce the suffering of families with Huntington disease. And that was one of the reasons I joined Teva, to be able to really fulfill that initial promise, because I realized quickly that Huntington disease has many features of that may be relevant also for many other disorders in the nervous system. And these include other disorders that have psychiatric changes with depression. They also have changes in cognition. And so Huntington disease represents a unique opportunity to give profound insights and to provide hope for families with Huntington disease, but potentially opening the door to understanding many other diseases as well of the central nervous system. Next slide. And so we started looking at pridopidine. Pridopidine was a drug developed by Neurosearch, and when I recognized that Neurosearch were having some difficulty, we approached them, and it was the first deal I did at Teva, was to bring pridopidine into Teva, because I had already seen some very interesting results from pridopidine. This was called a psychomotor stabilizer, and the results we had seen were already 2 phase 3 studies with significant effect on outcome, particularly total motor score. And what I was intrigued about with these data was that these patients who had pridopidine had some improvement in their motor function, the things that matter, that impact in the quality of your life, impairment of movements, coordination, being able to open a jar, do your buttons. All of these had shown some improvement, and the drug appeared well tolerated. There were no serious side effects, and also other symptoms, in particular chorea and depression, were not made worse. Next slide. So when we looked at how this drug worked, it worked to provide some balance for, we thought, to stabilize motor activity. When you look in animals, rats, for example, it was found to stabilize dopamine levels and also stabilize disorders where there were disturbances in motor function, either too much or too little. It brought it back to normal. So it acted as a motor stabilizer. And when you looked at the results, next slide, and you looked at the 2 phase 3 studies, and the first one on the left was the Mermaid Study. That was a phase 3 study with large numbers of patients, 437 patients, so very nice. And if you look at the total motor score shown in red on this slide, compared to the blue, which is essentially placebo, and going down, lower numbers means that total motor score is improved. What you see is that there's a significant difference between those who take pridopidine and those who don't, and this was after 26 weeks. But in this trial, and if you look at the heart study, which is next door, just on the right, it's a phase 2B study with 227 patients. Again, significant effects on the total motor score. But in both those studies, the defined score, the defined endpoint, the primary endpoint was, in fact, modified motor score that excluded, was a subset of the total motor score, and in particular also had chorea as a significant part of it, and excluded, for example, eye movements. And eye movements were important, but this drug has big impact on stabilizing eye movements. And so the primary endpoint wasn't met. And when I looked at this, if you go to the next slide, and you look at the pooled analysis from the phase 3 data, what you see is that here the total motor score was improved. Everything with the red circle indicates good, and minus means it's less severe. Eye movements were improvement, hand movements were improved, balance and gait, dystonia, all things. Dystonia means some stiffness with walking, some incoordination. And you can see all of those work very well. And if only the total motor score had been chosen as the endpoint, this would be a drug that's going to approval. And so when I looked at this and saw this data, I was able to persuade Neurosearch to essentially license this product to Teva, and immediately recognize that if we now go, the historic phase 3 trial was the modified motor score above and excluded some involuntary and eye movements, and the total motor score is now the primary endpoint of our trial. And you can see by what we've seen before that based on this, one has a reasonable expectation that we should see some effect. Now next slide. So we went to the regulatory authorities with this data, and they told us that if we are able to achieve some rigorous endpoints, that they would consider allowing us to submit this for approval at the end of this study, which would be sometime in 2016. And this is a study, what was also interesting is that if you looked at the prior study, and Spiros is going to go to much more detail, but we also would recognize that there were much better doses that we could use and higher doses that might show better effects. And so we've now, this is a large study of 550 patients in 12 countries around the world in 54 sites. So this is really a very significant large study, and essentially if the results from the current study meet the criteria outlined by the FDA, we would expect to submit in 2016. Now next slide. But the interesting thing for us, at the time Neurosearch was a very small biotech company, didn't have a lot of resources, and I was most interested to learn more about what this pridopidine had done, because these results were quite remarkable and looked very encouraging. And so we have now learned that not only does it provide motor stability, but it also changes various things in brains. And we've been looking, obviously, in animal studies. And what we have found that not only does it stabilize dopamine, but it also has some neuroprotective effect. It looks like it protects neurons from further degeneration. And also it has impacts on various neurotransmitters. These are chemicals that transmit essentially impulses throughout the brain and are important for thinking and feeling. And these also improved some of these, which suggested to us that this may have impact on, there may be some broader effects on other features of Huntington's disease. And so this, this made us think that, in fact, this study may have a broader perspective beyond just what had been looked forward before. And as Spiros will tell you in a moment, because of that, the study has a lot of other endpoints that are very important. Studies that affect cognition, studies that affect mood as well, studies that affect the quality of life, because there was some hope from this early data that we're seeing now that this drug may have broader effects. Next slide. So what we're looking at here is that a drug that we've already shown has effects on motor function, but also had potential for neuroprotection, changes in cognition. And with this, we really are thinking that this may not only have some symptomatic effect, but could theoretically have some effect to change the course of the illness. And wouldn't that be exciting? And we wanted to be able to look at that. And because these studies are ongoing right now, we've also created an extension study that goes on a year that will give us some indication as to whether this drug is having any effect on the progression of the illness. Next slide. Now, what we do know is that essentially the neuroprotective effect in this slide, and if you just look in this slide, essentially low is not good, and what you can see here with the red is that when you look at the second lane there with pridopidine with 200, you see it shows some really, as you go up there, it shows protection. And this is seen throughout, and very similar seen to like a vitamin dose for the brain, BDNF, which essentially is good for the brain and good for neuronal cells. And these are cells in a culture situation, and you can see that this drug has a dose-dependent effect. And this is why we also recognize we wanted to look at numerous doses, because you may see some of these effects at higher doses, and we needed to look at this. And furthermore, next slide, when you looked at the areas of the brain that were involved, the areas of the brain were involved, regions that really were important. The striatum, which is the area of the brain, the core central part of the brain where patients and people with Huntington's disease have nerve loss. And the cortex, which is the part of the brain that's above that, over the covering and important for many aspects of function. The hippocampus, important also of thinking and emotion. And so the increases in neurotransmitters occurred throughout the brain, and this is the reason why for some people going through the study now, it's quite exhausting because we do a lot of things, and this is the way that studies are done. You have to show that they're effective before the regulatory authorities will allow us to be able to provide these drugs to patients around the world. So the new data, last slide before I'm going to hand over to Spiros, is that there is really a chance that pridopidine may have a broader effect beyond symptomatic relief. We don't know that at all now. We don't have data in humans, but we're hoping to find that. And the phase 2-3 study that we're doing, and phase 2-3 means that it's a late study. Drug development goes through 3 stages, phase 1, phase 2, and phase 3. This is a phase 2-3 study, and essentially it's been modified to include some of these additional exploratory endpoints that allow us to know whether this drug really has any effect beyond the symptomatic effect in motor function. So you can see why I'm particularly thrilled to be part of this opportunity to be able to influence the commitment to Huntington's disease, and this represents a long cycle from the days in South Africa and with Marjorie Guthrie in the early days where the horizons for therapy were very far away. And we are now in a place of really a tremendous hope. I think the darkness is replaced by hope and light. And at the end of the tunnel, we are seeing this light now, not only with this drug, but other trials that you're hearing about through the webinars, so wonderfully supported by HDSA. And I'm going to ask Spiros now to continue to tell you much more detail about the pridopidine study or the PRIDE-HD study. And I can tell you, I feel very proud to be part of this particular study, and so pleased that Spiros is here to help navigate and help provide the leadership to bring the study to completion. So Spiros, over to you.
Thank you very much, Michael. Again, my name is Spiros Papapetropoulos. I'm a movement disorders neurologist. I trained in Greece in the United Kingdom before joining the University of Miami. I was faculty, neurology faculty at the Department of Neurology at the Division of Movement Disorders, and I had the privilege of meeting patients and families with movement disorders, including Huntington's disease patients. As Michael and George mentioned, I recently joined Teva to lead a group that is responsible for developing the next generation therapeutics for neurodegenerative diseases. And both Michael and I share the same vision about Huntington's disease and excitement about this program. Before moving forward, I like to thank the HDSA for the invitation to present, and most importantly, today's participants for taking the time out of your busy lives, busy schedules to attend this webinar to find out about Teva's Huntington's disease program and the pridopidine dose escalation in HD study, also known as PRIDE-HD. I just want to briefly recap what Michael said. Pridopidine is an investigational drug being studied for its efficacy and safety in Huntington's disease. It is believed that its effects on some of the symptoms of Huntington's disease are dependent on dopamine. Other neurotransmitters such as histamine and acetylcholine may be implicated. Dopamine is a substance that is naturally made in many areas of the brain that affects how people move, think, and behave. As Dr. Hayden described, in a pooled analysis of clinical studies completed today with pridopidine, pridopidine was found to have efficacy on motor aspects of Huntington's disease, and further investigations are being performed to look into other non-motor or other features of Huntington's disease, including behavior and cognition. So the promise is there. We're just testing the hypothesis at this moment. In our clinical development programs at Teva, patient safety is always our priority. To make sure that our investigational drugs are safe, we compare rates of adverse events in patients taking active drugs to those of patients on placebo or a sugar pill. If these rates are comparable, the interpretation is that the investigational drug is safe and well tolerated, and development usually and typically continues. I would like to direct your attention in the columns that are in red circles, and more importantly, at the percentages of patients that experience AEs. This data is taken from the two completed studies presented by Dr. Hayden called HEART and Mermaid HD. You will observe that across the board, there are similar percentages between the pridopidine and placebo arms. By looking at this data, our conclusion is that pridopidine up to doses of 45 milligrams twice daily has been safe and well tolerated with similar levels of adverse events to those of placebo, therefore enabling current and future investigation of similar and higher doses in patients. What is our goal with PRIDE-HD? Our goal with PRIDE-HD is to test higher doses than previous study to assess the effect and dose response of pridopidine of movement, thinking, and behavior in patients with Huntington's disease. This is really what we're after. I've decided not to go into great detail around the individual measures that we're using, but as Michael alluded to, we are using a variety and a battery of tests that are administered by neurologists, others that are patient-reported tests, others that are reported by caregivers that capture the variety of symptoms and signs of Huntington's disease. The actual name of the study, the actual study title, is relatively complicated. I will take a moment to read it out so you can appreciate why we chose to use an easy-to-remember acronym as PRIDE-HD. So the study is entitled a Phase 2 Dose-Finding, Randomized, Placebo-Controlled, Double-Blind Study evaluating the safety and efficacy of pridopidine, 45 milligrams, 67.5 milligrams, 90 milligrams, and 112.5 milligrams twice daily versus placebo for symptomatic treatment in patients with Huntington's. Wow, this is a mouthful. This is a placebo-controlled study with participants randomly assigned to receive capsules containing 1 of 4 doses of pridopidine or placebo. So participants actually have 4 out of 5 chances to receive pridopidine. The placebo capsules look like pridopidine but have no active ingredient. They're also known as the sugar pill that I mentioned earlier. Everyone will receive the same number of capsules that are white, and neither participants nor study doctors will know which group subjects in the study are assigned to. And that's the whole notion of a controlled, double-blind, randomized, placebo-controlled study. Going into some more detail about our study, there are 400 people that are planned to enroll across three continents in 54 sites in Austria, Denmark, Germany, Italy, Netherlands, Poland, Russia, UK, Australia, Canada, and the US. The backbone of this study design is presented in the schematic on the bottom of this slide. This is a 12-week period to ensure patients meet participation criteria, followed by a 2-week period during which, in some patients, investigational study drug is dosed gradually, increasing to levels that have not been studied before. So previous studies, such as Heart and Mermaid, studied doses up to a maximum of 45 milligrams twice daily. In this study, the doses are going to increase substantially, up to 112.5 milligrams twice daily. Our hypothesis is that higher doses will be as safe as lower doses, but will have better efficacy. This is at least what the preclinical data is suggesting. The total time participants will be on investigational drug or placebo is 52 weeks, or about 12 months, during which time they will visit the study site 11 times. Subjects will also receive phone calls from qualified study personnel to ensure that during and immediately after the gradual increases in doses, there are no adverse drug events that could compromise participant safety. Participants also need to visit the study site for one last time during a follow-up visit 2 weeks after stop taking the investigational drug or placebo. This discontinuation is required to collect information on what happens when a patient discontinues pridopidine. This information is required for approval from health authorities. That is the reason why we've implemented it. For someone to qualify for PRIDE-HD, they must meet a list of 12 inclusion and 18 exclusion criteria. I'm not going to go over this exhaustive list, but I'm going to highlight the most important ones that are age 21 or over, with disease onset over the age of 18, participants should have a caregiver willing to attend study visits with them. They should not have no history of significant heart disease at screening, should be in general good health, should have no history of epilepsy or seizures within the last 5 years, should have not been treated with tetrabenazine, also known as mitomine or xenazine, within 6 weeks of study screening. There are additional criteria, many of them, as I mentioned, that should be met in order to qualify for the study. The study physician will discuss these with patients that decide to participate in great detail. Prior to participation, patients will be asked and required to sign an extensive informed consent that explains the study design, the risks and benefits of pridopidine and the study in general in great detail. But what happens when a patient completes a PRIDE-HD? Patients that successfully complete PRIDE-HD will be eligible to enter Open PRIDE, the so-called open label extension of PRIDE-HD. But what exactly is an open label extension? It is typically the long-term extension of a double-blind study that are called open label because during this open label extension there is no placebo arm. Both patients and study personnel know that they're receiving the investigational product. That's why we're opening the label. It's no more double blind. The dose that patients will receive in Open PRIDE is 45 milligrams twice daily until PRIDE-HD is completed. After data review from PRIDE-HD is conducted, doses may increase. In terms of complexity, Open PRIDE will be simpler. It will be like PRIDE-HD, but a lot simpler, with less frequent visits and procedures, because it's going to be a long study. Open PRIDE will continue until either marketing authorization is granted in the US and Europe, or the program is discontinued due to a failure. Basic reasons is no efficacy, for instance, after the double-blind part of the study. You can find more information about Pride HT on HTSA's website or by calling HTSA at 1-800-345-HTSA. And I would like to thank everyone for your participation, for your attention, HTSA for your invitation, and Michael for providing a great overview of the program. I'm really looking forward to working with everyone that is involved in Huntington's disease research, and I'm looking forward to great years of significant success that will improve patients' lives. So this presentation is now open to questions. Thank you very much.
Thank you so much, Spiros, and to Michael for that really great presentation. So as a reminder, folks online, if you have any questions for Michael or Spiros, please type them into the questions box now. There's a number of questions, Spiros. First one is, can you talk about how pridopidine compares with Auspex, SD809, and Xenazine?
Absolutely. I'll be delighted to do that. The Auspex's version of tetrabenazine is a promising, appears to be a promising improvement over the standard tetrabenazine. It uses a proprietary platform that Auspex has developed, and based on the publicly available information, it is expected to provide effects similar or even better to tetrabenazine. As it is well known, the effects of tetrabenazine are primarily on the chorea symptoms of Huntington's disease, and as opposed to pridopidine that has shown promise in a wider spectrum of symptoms and signs. So it is going to be really interesting to observe the totality of data that Auspex will present, and I'm sure that it's going to be, if successful, it's going to be a great addition to the current treatment paradigms.
Great, thank you. Another question, is it an interesting question coming all the way from Germany, and it relates to just the name. There's, I guess, a little confusion about the name pridopidine versus maybe people having known it as Huntexal. Can you just clarify for people the differences in the names that this drug has had in the past?
Yes, absolutely. Thank you very much for this excellent question. It is indeed confusing. However, the real name of this drug is pridopidine. Huntexal is a term or a name that Neurosearch used, and in the future, the name of the ingredient is going to be referred to as pridopidine. If successful and after commercialization, and depending on regions, this may change, but for the time being, there should be consistency in naming. So Huntexal, pridopidine, and ACR are the one and the same drug, and it's pridopidine. So I do appreciate the opportunity to clarify this.
Yeah, and I'll just add to that, it is confusing in the industry because it's a little bit like naming your child, and every company wants to name their child in their own way. So Huntexal was named essentially by Neurosearch, but you also have to get the appropriate, Teva, of course, it's a different company, wouldn't use that name, but the naming has to be approved by the regulatory authority, so it has to go through a process of naming, and this is only relevant for when it would get to the market, and pridopidine was never approved till now, and so it will have a name when it gets to the market, but these are all the same drug. There's no modification, but I can understand how the confusion arises.
Thank you, guys. Another question, just trying to get clarity for, I guess it's a caregiver or a physician saying, how do we get, what's the best way to get our HD patients into this study? What would you suggest is the clearest path forward for them to start the process?
So let me just answer first, and then I'm going to hand that over to Spiros. But I'd say, what would be important is for those people who feel that they may be eligible. They'll, of course, have to go through a process. There are a large number of sites, not only in the U.S., but in 12 countries, Canada and various parts of Europe. And I would say one way to do this is to look at HDSA, call HDSA, 1-800-345-HDSA, in an effort to ask what would be the closest clinical trial for this, and then for people to inquire. I think Teva ourselves, we're also available. I mean, people could write to Spiros, and Spiros could also help direct people to the right place to see whether they're eligible. And we want to encourage. I mean, we would like to make sure that the study is fully enrolled and reach the numbers that are necessary so we can know as soon as possible whether this is a drug that should be made available to everybody. And we don't know that yet, so that's the reason for the trials. But Spiros, let me hand over to you.
Sure, so absolutely. Thank you, Michael. This is another great question. For this particular study for PRIDE HD, it's important to enroll it quickly so we can get the necessary answers that we're looking for. We're optimistic, and we partner with organizations like the European Huntington's Disease Network, HDSA. The Canadian Society knows about this study, so your best resource is your local society and patient advocacy group. And the second resource, or the next best resource, as good as the first one, could be Teva, myself personally, and the whole HD team. So we're looking forward to receiving any inquiries you may have and helping you navigate and find the right site for participation. But it's important to stress that the study is ongoing. It is enrolling, so the opportunity for participation is going to be around for the next months. Hopefully, not too long before we can safely complete enrollment and have results that will enable submissions.
So, and I think if you do call HDSA, HDSA will have contacts and coordinates for Spiros, and so you could make the contacts that way so Spiros can help you too. We want to be also very available for you, but I think HDSA may be a good place to start.
Absolutely. Thank you, guys. Couple questions. This is a quick one regarding the Open Pride study. They just wanted some participants wanted clarification that the Open Pride study, the open label, will only be available for former Pride HD participants. Is that true? That's the case, correct?
Yes, that's correct.
Okay. If people wanted to clarify that. Question about the neuroprotective effects or potential neuroprotective effects of pridopidine. Are there any thoughts on the possible mechanism of action that pridopidine may be having to exhibit the neuroprotective effects?
Well, we do have some leads. We're not quite ready. It's sort of like research data that needs to be further proven, but it is. We have uncovered a pathway for this drug that looks very intriguing and is very consistent. It's a pathway that's been known to have big impact in the brain on some of its neuroprotective and cognitive effects. So it is very logical it's very congruent with what we know, but I wouldn't want to talk about it until we've got further proof of this. And the way we're proving these things is by using various neurons that we're able to create or get, and usually from animals, where you can actually knock out a certain gene and see if it loses its effect. Then you know that effect was critically dependent on that gene. And these studies are underway. And so until we really know for sure, but the early data looks very intriguing relative to pathways that we do understand. And already there's been lots of work on pathways, not with our drug, but with others related to these pathways. And these pathways, when activated, have been shown to have effects not in Huntington's disease, but in other illness, improving cognition and improving neuropsychiatric effects, and also shown to have neuroprotective effects. So this is, we ask you just to keep, keep posted to this, and once we know for sure, we will want to share it, but it's still a little early.
Thanks, Michael. This is a great question and a good observation from the community that according to the HSG website, and I'll admit that it's also the HDSA website, that we initially advertised that the trial PRIDE-HD is running 26 weeks. Has this changed? And also, second question, for people who've already started the trial, the PRIDE trial, if Open PRIDE is not approved until September, will they have to go off the medication before starting the open label trial?
Well, that is another very important question. I appreciate the opportunity to comment. The PRIDE-HD study was recently amended, and the amendment includes one of the changes, and the major change was the extension of the study from 26 weeks to 52 weeks. So yes, that is a change that was recently implemented, and there is a very robust scientific and clinical development rationale that is centered around safety and ensuring that pridopidine is safe throughout a duration of 52 weeks. So that was the main driver behind having this amendment. And the important element of the amendment is we're going to be able to compare the different doses of pridopidine, the safety of different doses of pridopidine to the safety of the placebo arm and make a meaningful comparison. The second reason why an amendment was implemented was the efficacy component will be now explored further. So we're gonna have a better opportunity to look at the sustainability of effect of different doses of pridopidine. So these are a couple of main reasons why this amendment was implemented. For the subjects that exit PRIDE-HD, we're taking all the necessary steps to make different options available to them depending which geography they reside. So there's not a single approach for everyone. The single approach for everyone will eventually be Open PRIDE, which will be available in August, September time frame for everyone that is exiting PRIDE-HD. But until then, we have options through Open Heart in the geographies that Open Heart is running, and we have also initiated efforts for compassionate use program, which is going to be available at a case-by-case review. So I don't know, Michael, if you have any other feedback for this very important question. Okay.
Great, thanks, Spiros. A couple questions coming in, and I'm not sure if you, at this early stage, you probably don't have an answer for them, but I want to make sure that they're they're asked. And people have questions about the affordability of the drug if approved. Has there been any discussions on what the price of pridopidine would be if it's an approved medicine, and and whether Medicare for it?
Well, this is going to be an intense discussion with the local payer systems. Typically, this is set post-approval, and it's dependent on the result. So it's a very complex process, and it uses local practices and benchmarks, and there are a lot of different stakeholders involved. In terms of an exact pricing, it will depend, and again, on the geography, and it's going to depend on the outcomes of the study and the outcomes of different regulatory with different discussions with payers.
Thanks, Spiros. Just one question was just coming in about if they can get a copy of these slides, and I just want to remind everyone that we will turn this, we are recording this, and we'll turn this presentation around and make sure it's posted on our website, hopefully by the end of the day, if not by first thing tomorrow morning. And another final question was, were there any physicians or clinicians in Puerto Rico that were participating in this trial? And I think I can at least try to handle that. And I do have a slide prepared that shows the different U.S. sites if people were interested. There's, I believe, 10 right now. You can see them hopefully on my screen at the institutions in the cities and states and where the trial is. So those are the 10 trials if you're interested in connecting with them. If you have any questions, you can, as Spiros and Michael said, call HGSA or call the Huntington Study Group assisting with this trial, and both, I'm sure, will be happy to put you in contact with the site coordinator at the nearest center. One final question, Spiros, is maybe you can comment, there are a lot of site visits for that require caregivers to attend over the course of a 52-week study. Is Teva providing any kind of travel reimbursement to families to help offset the costs associated with participating in this trial?
Very good question. Yes, we are making all the necessary steps towards securing travel assistance, and you will be able to find out more once you contact us or the study site. So the answer is yes.
Great. Well, thank you, Spiros and Michael, for a great presentation. There appear to be no other questions. So, like I said, if you have any questions, don't hesitate to contact HGSA or HSG or Spiros. We're happy to assist you if you're interested in participating in this trial or others. So thank you again, and we hope to see you or have you participate in our upcoming webinar on March 19th. Thank you so much, everyone.
Thank you.
Thank you. Bye.
Bye-bye.